Hi everybody, today we are going to discuss the collagen. Collagen is the most abundant extracellular matrix protein. It is a structurally very important protein. It is found in connective tissues. Collagen, it is made up of 3 polypeptide chains. It shows like a helical model. The collagen is more than 25 types so far identified. All the types are mentioned as a Roman letters. Clear? The collagens, where it is synthesized in fibroblast, muscle cells, and epithelial cells also can produce collagens. Clear? So the collagen, it is synthesized in fibroblast, muscle cells, epithelial cells. What is the main functions of the collagen? So collagen, it can give main support to the all organs. So that is the main functions of collagen. And then it can useful for this, collagens are very useful for cell alignment, so that cell anchoring is possible. So that is very important. Clear? So that is a collagen. Okay. So the collagen means it's a Greek word which means the substance will produce glue-like substances. So that is called as collagen. The collagens are, the shape is like a triple helix. It is made up of 3 polypeptide chains. Clear? So it is contain 1000 amino acid, means more than 30% are glycine amino acids. Glycine amino acids. Clear? The structure shows like okay, this is glycine, X, Y, glycine, X, Y, like that. Every number 3 is glycine. Clear? X may be for any one of amino acids. Any one of amino acids. Y, it is mainly for hydroxylation, hydroxyproline. That means hydroxy amino acids are present here. Clear? So that is a very important. Why the more than 30% of the glycine is present? Because glycine is being very small in size. Molecular weight is small molecular weight. It can fit into the crowded interior of collagen. That is one reason. Second reason, okay. First reason, it's a small size. It can fit inside of the crowded area of collagen. Okay. Then second reason, it can allow shallow grooves. If it allows the shallow grooves, means then one polypeptide chain can overlap another polypeptide chain. The shapes like this shape. Clear? So that is the reason more than 30% of the glycine is present in the collagen. That's a very important multiple choice question. Clear? Now we will see. Okay, you know where it is synthesized. Now we will see how it is synthesized. Clear? Before that, it is contain 3 alpha chain. Okay, alpha 1, alpha 2, alpha 3. Or else, say for if it is alpha 1, all the 3 are alpha 1 chain. All the 3 are alpha 2. All the 3 are alpha 3. That means homo trimer. Suppose if it is contain alpha 1, alpha 2, alpha 2, or else alpha 2, alpha 1, alpha 1, or else alpha 3, alpha 1, alpha 1. That means this is called as heterotrimer. Heterotrimer. Clear? Okay. Now we'll see where it is and how it is synthesized. Clear? This is one polypeptide chain. It is synthesized from ribosome. Proteins are synthesized from only ribosome. Clear? And it is contain some different signal. So this is called as signal peptide. This is called as signal peptide. Clear? The signal peptide, this area is signal peptide. Clear? Now what will happen? This is called as pro alpha chain. So it is coming from ribosome. It is for the movement purpose. Then it is reaching into the lumen of rough endoplasmic reticulum. After reaching the lumen, the signal peptide is cleaved by the enzymes called as peptidase enzyme. Then the signal peptide will be digested by lumen of rough endoplasmic reticulum. Then you can get N terminal, C terminal of pro alpha chains, because like this the pro alpha chain is coming, 3 molecules of 3 polypeptide chains are coming. So that's why I mentioned pro alpha chains. Clear? The 3 pro alpha chains. So 3 molecules of polypeptide chains are coming, and how it is holding? It is holded by disulfide linkages. Disulfide linkages. The disulfide linkages is produced by sulfur containing amino acids, cysteine, and methionine. The 3 amino acids are responsible to hold these polypeptide chains. Clear? This takes place in the lumen of endoplasmic reticulum. Clear? Now the lumen of endoplasmic reticulum is produced one more reactions. Clear? That is called as hydroxylation reaction. Hydroxylation means, you know already how many amino acids are contained hydroxyl groups, that is serine, threonine and tyrosine. Only the 3 amino acids are hydroxy amino acids. The 3 amino acids is not sufficient, because this is going to make like a rope like structures. Clear? That is why the shape that I already told you, the collagen shape is like a rope like a structure, or hair braid shape. So that means it is required more amount of hydroxy amino acids. So that's why they are lumen of rough endoplasmic reticulum introduce 2 more amino acids. The 2 more amino acids, say for example, that is called as proline and lysine. The 2 amino acids are undergo hydroxylation reactions to produce hydroxyproline, hydroxylysine by the enzymes called as, proline means prolyl hydroxylase enzyme. Lysine means lysyl hydroxylase enzyme. Then it can produce hydroxy amino acids. Clear? So the 2 amino acids are introduced by lumen of rough endoplasmic reticulum. Okay, hydroxylation reaction. For this hydroxylation reactions, okay, that vitamin C is important as a cofactor. Clear? Suppose vitamin C is absent, then what will happen? There is no chance to formation of hydroxylation reactions. Clear? So the hydroxylation means compulsory vitamin C is required. Clear? Now the 3 chains. So I written only chain. Here is I am written chains, because the 3 chains are joined by disulfide linkages. Clear? Then the structures, the shows like this shape. It is contain disulfide linkage. Now the structures, how it is joined like a hair braid shape, because of only hydroxylation. More, you know already primary structure of protein. Primary structure of protein, this is primary structure of protein. Secondary structure of protein means this is secondary structure of protein, because secondary structure of proteins contain hydrogen bonds. So the more hydrogen bonds will form like a zigzag structures. Clear? Like that you have to remember the secondary structure of protein. Okay, that is called as, this is pro alpha chain is converted into pro collagen. Clear? Pro-collagen only you can see the shapes like a twisted shape, or else hair braid shapes, or else rope like structures. Clear? Now the pro-collagen, it is going to the Golgi complex. Golgi complex. Why it is going to the Golgi complex? Here only it is G for glycosylation. It is glycosylation is going to occur here. Glycosylation. Here is glycosylation means glucose or galactose are attached into only lysine amino acid, not with any other amino acids. Clear? So glycosylations occurs only, that's a very important MCQs. Glycosylation occurs only in lysine amino acids. Okay. What is the uses of glycosylation? Because the glycosylation only can give the structure will be assembled by the collagen structures are assembled by only glycosylation. Okay, later I will explain you why it is attaching only in lysine. Because of the lysine, then only the lysine will involve cross linkages. So that is the main purpose. The glycosylation takes place in the Golgi complex. First of all, after coming from this ribosome, it is going to the lumen of rough endoplasmic reticulum. Then it is going to the Golgi complex. The Golgi complex means the 3 stages are there: cis Golgi, medial Golgi, and then trans Golgi. After the coming out of the trans Golgi, then it is getting glycosylated. Clear? Now the pro-collagen, it is converted into tropocollagen. It is converted into tropocollagen. How? Here now see here, then after this come out of trans Golgi, then it is going to the extracellular matrix. Extracellular matrix. Now in the extracellular matrix, there is a 2 important enzymes are available. That is called as, okay, this is called as N terminal. This is called as C terminal. So N terminal, this area is completely bulged because of disulfide linkage. It cannot be joined. It cannot be intertwined. Okay. So that is why this N terminal pro-collagen propeptidase enzyme will remove the bulged area, and this area, okay, C terminal pro-collagen propeptidase enzymes will cleave this one. Then you can get only this part after removal of the bulgy part, that is called as tropocollagen. This is called as tropocollagen. Clear? The important 2 enzyme, N terminal pro-collagen propeptidase, C terminal pro-collagen propeptidase will cleave this disulfide linkages. Clear? Then you can get tropocollagen. Now the tropocollagen is converted into collagen. This reaction takes place in same extracellular matrix. This is called as cross linkages. Cross linkages. Clear? What is the meaning of cross linkages? Cross linkages are 2 types. The type 1, 2 types. The first one is called, that is called as Sif base. Sif base. Another one is called as aldol condensation. Aldol condensation. Okay. Now what is the meaning of aldol condensation? We will see now. There is lysine amino acid. The lysine amino acid and hydroxylation amino acid. Hydroxylation amino acids undergo oxidative deamination. Oxidative deamination by the enzymes called as lysyl oxidase enzyme. Then it forms these amino acids. Lysine amino acid, hydroxylation amino acid is converted to allycine. Allycine means it is made up of only, it is contains only aldehyde group. Clear? So for this one, lysine and hydroxylysine undergo lysyl oxidase enzymes by the process called as oxidative deamination. We are adding the oxygen molecule to removal of amino group. Clear? So that is called as oxidative deamination. Then it forms allycine. It is contain aldehyde group. So what is the 2 types I told? The 2 types means Chief base. Chief base means I told you it can produce aldehyde. This aldehyde combined with another polypeptide chains of amino group is joined aldehyde amino group. That is called, this is one chain, this is another, this is another. So this one aldehyde, here is amino group. Both are joining. That is called as cross linkages. So that is called as chief base. Okay, this is one type. Another type is aldehyde condensed with another molecule of aldehyde, another polypeptide chains of aldehyde. Here is aldehyde group. Here is aldehyde group. Then this aldehyde and aldehyde, this both are joined. That is called as aldol condensation. Then you can get more cross linkages. The tropocollagen is undergo cross linkages. Then you can get proper collagen structure. The collagen structure is it's made up of like this type shape. Then it is contain more cross linkages, become very rigidity. So that is the steps of how the collagens are synthesized. Now we will see what is the abnormal of the collagens. Abnormal of the collagens, because of only there is any mutations occur in gene, any one of the genes, it is any mutations occur, that is called as abnormal of collagen. First one is called as osteogenesis imperfecta. Osteogenesis imperfecta. Osteogenesis imperfecta means there is any mutations occurs exactly in type 1 collagen. Mutation in gene type 1. So that is called as osteogenesis imperfecta. The osteogenesis imperfecta is otherwise called as brittle bone disease. Brittle bone disease. That means the bones are become deformity, bones are become weakness. Okay, bones disorder. That is called as osteogenesis imperfecta. Along with this one bone and your deafness also is possible. Deafness and blue sclera also possible. Blue sclera and dental abnormalities also possible. So that is called as osteogenesis imperfecta. Okay, osteogenesis imperfecta also having some different types, but only we need only very important one is this is affect in the mutation gene in type 1 collagens. Clear? So that is called as osteogenesis imperfecta. Second one is called as Ehlers-Danlos syndrome. Ehlers-Danlos syndrome. Clear? This also the mutations are occurs, especially in gene 1 and 3. Especially gene 1 and 3. Okay, the mutations. Gene 1 and 3. And here is one more thing, that is a completely absence of a lysyl oxidase enzyme, lysyl hydroxylase enzyme. Hydroxylase enzyme. These enzymes are absent or defect. Then it leads to what will happen? These enzymes are absent means what will happen? It is not. It will be like this shape. It will be like this shape because cross. That means hydroxylase means it can interconnected. But here is hydroxylase enzyme absent. Then what will happen? Hypermobility, hyperextensibility, or sometimes it can leads to disease that is called as rupture of our vessels also is possible. That is called as Ehlers-Danlos syndrome. Clear? Then next one is called as Alport syndrome. Alport syndrome. This is completely affect our kidney. This is completely mutation genes occurs in type 4. Type 4 means that is called basal lamina. Okay, mutations gene basal lamina. Then what will happen? It can mainly affect our kidney. Then starting to that is called renal damage. If it is more renal damage, it can lead to hematuria. If it is continuous hematuria, then finally you can get renal failure condition. So that is called as Alport syndrome. Clear? Then next one is called as Menkes disease. The Menkes disease, that is also Menkes disease. Here is poor or very less amount of absorption of copper from the intestine. So that is called as Menkes diseases. Clear? And this is also mutation occurs and 7 mutations gene. Here is one special is there, that is called ATP 7 gene. is affected, completely getting mutation, and poor absorption of copper. Clear? That is called as Menkes diseases. Next one is called as 1, 2, 3, 4, 5. 5 is called as chondrodysplasia. Chondrodysplasia also, it is also mutations occurs in especially type 2 genes. Type 2 mutations gene in type 2 collagen. Clear? So the chondrodysplasia will come. Then what will happen in these conditions? In the chondrodysplasia, that means the person will not grow. Okay, the appearance shows like a midget, or else otherwise called as dwarfism. Clear? Then sixth one, that is called as epidermolysis bullosa. Epidermolysis bullosa. Epidermolysis bullosa means some skin having, it can produce like a blister. It can produce blister. This is especially the same mutations. The mutations of gene in type 7. What will happen initially? You can get like a blister formation. Then the person will be scratching. Then leads to like a epidermolysis bullosa. It shows like a resemblance of psoriasis. Clear? Then last one is that is called as scurvy. You know very well scurvy. Completely absence of vitamin C. Vitamin C absent means what will happen? Hydroxylation reaction is not possible. So that is, it leads to scurvy diseases. Clear? Then if it is a scurvy disease means a spongy gums, loosening of teeth, okay, bleeding of gums. So that is called as scurvy disease. So this is called as collagen. How it is synthesized, where it is synthesized. Clear? And what is the abnormal of collagen. That's all. I hope you understand. Thank you. Bye. இருபத்தி ரெண்டு முடிச்சிருக்கேன்.